minutes, please. You know, you have actually given us.
All right. Good evening once again, the audience, the panelists, all invited guests. We are here to share with you a platform that we just created, AMR X Hub, which is a platform for aggregating antimicrobial resistant tools and resources. It's our team. Launch on the 15th of August this year. These two was developed to address one of the critical challenges we face nowadays, which is antimicrobial resistance and tool aggregation. We found that this platform was inspired by the demand for a centralized repository for antimicrobial resistant tools and resources, which was scattered across different websites and platforms all over the world. And hence it was suggested by the Director of Science, Helix Biogen Institute, to support the One Health approach that AMR demands, connecting human, animal, and environmental health stakeholders. Before AMR X Hub, we had information fragmentation, lots of research time wasted, no quality signals, and access barrier due to poor indexing or regional constraints. But with AMR X Hub, we have a centralized discovery, rapid access to such material, a rich metadata that is constantly being updated, and global access. This is a glimpse of what AMR X Hub currently looks like, still in development. The information you can see before you shows that in the span of 30 days, which is between the 15th of August to 13th of September, AMR X Hub reached 14 countries. The top being the United States, Nigeria, Canada, France, and Germany, showing there is a demand for it internationally and locally. And our search engine performance was, on average, 5 times better than every other platform that started in this niche. We have some goals moving forward, such as our short-term goals, including operational restoration, automated link monitoring, enhanced searching, and user contribution to tools and resources. Our midterm goals include front-end migration, community features, and partner portals, while our long-term goals include the hosted tool environment, federated indexing, AI-assisted curation, sustainable model. Our current challenges and limitations we face include link runtime maintenance, because not all data online is updated, variable metadata quality and coverage gap, which requires a lot of staff to fill in. While other challenges include financial sustainability, curation workload, and the fact that we don't have an environment to host any of these tools yet. And these endeavors are quite expensive. Now, for every tool and material we provide on our platform, we always provide information about them in the description to inform the user about the license requirements and others. In conclusion, AMR X Hub is a platform that brings together dispersed antimicrobial tools and resources from all over the web. We would like to acknowledge our Director of Science, Helix Biogen Institute, Associate Professor Oladipo Elijah Kolawole, and all the staff for their support. Our selected references. Thank you for listening.
So thank you for your presentation. So briefly, the presenter, their team established an existing, AMR, repository, using data from Europe, from USA, to really see how to gather the fragmented resources, data on AMR. So that's what he has presented on. So I call on the second presenter, that's Oladapo Dihai. 5 minutes, please.
Good afternoon. Even though I'm not Oladapo Dihai, but I'm part of the team that worked on that. I'm Akinshola Viho. So this is a machine learning approaches for early detection of non-communicable diseases in low resource settings. Yes, I know that for biomedical research, the interest is basically and usually on infectious diseases. But really, if you look at the data, we have more mortality for non-communicable diseases than infectious diseases. You can also always see that. But the hype is always on infectious disease, maybe because of the fact that it's an in thing and all that. So this is is an aspect of, so the non-communicable diseases we look at are basically diabetes, hypertension. They are the leading causes of morbidity and mortality globally. And in Nigeria, there are many cases that remain undiagnosed. Many times the non-communicable diseases are seen like they are called silent killers because you don't know them until there is already a complication. So, and at that time, it's too late, sort of. And because non-communicable diseases, they don't go. So they are chronic in nature, and once people have it, they continue to manage it. They are just managerial management diseases. So early identification is a major thing. And so we looked into models, AI models, and machine learning models. So we developed predictive machine learning models for early detection of major NCDs. We compared the performance of different algorithms. So we used 3 statistical algorithms, logistic regression, random forest, and gradient boosting. So we evaluate the feasibility for low resource NCDs. Next slide. Now, the data. So the data is going from health record of patients with NCDC from tertiary hospital in the southwest. So the NCD status. So we look at independent variables. The independent variable is the NCD status. So why the dependent variables are the age of the patient, body mass index, blood pressure, glucose level, and lifestyle indicators. These are basic indicators for the non-communicable diseases. The age influences this, the BMI and the blood pressure level, the glucose level, and lifestyle. So the models use 3 models, logistic regression, as I've said. Then we did evaluation of the performance. We assessed that using the area under curve, that's the AUC, the area under curve, sensitivity and specificity to ensure the sustainability for deployment for low resource clinical settings. Many times when you need to, because usually it will have been, it is when people are already showing symptoms, that's when they go to the hospital for hypertension and for diabetes, usually. So in a bid to be able to see, normally taking the vital signs like age and all of those things, so that we can be able to predict. That's what we are using the models to do. So we implemented with Python. Next slide. Now, this is the result. Now, if you look at this, the logistic regression for accuracy is 75.2. So the gradient boosting have the highest accuracy. So, okay, so we see that accuracy. Now, random forest and gradient boosting, they are example of the ensemble models. So you can have random forest and gradient boosting. So those are the examples of the ensemble models, and you see that the ensemble models, they did better than the regression models. Regression is, I know many of us should be very conversant with regression. So next slide. Okay, so this is the essentially the second graph that shows the features of importance which is in the ranking. The systolic BP, the BMI, the glucose, age, and lifestyle. These are major determinants for whether somebody will come up with an NCD, either diabetes or hypertension. So now that you see that the BP is a very, very important precursor to somebody developing non-communicable diseases, especially diabetes, either diabetes or hypertension. So that's what we can see from that. Okay. The conclusion. So machine learning models, particularly random forest and gradient boosting, they demonstrated strong potential for early detection of non-communicable diseases in low-resource hospital setting. And Okay, the gradient boosting model offers the best balance between sensitivity and specificity, making it suitable for clinical screening and digital health integration. This finding shows that data-driven approaches can complement existing diagnostic systems and strengthen healthcare capacity in resource-limited environment. Thank you.
Thank you for your presentation. So briefly, the researchers, they use machine learning to predict non-communicable diseases using age and glucose level and others as their predictive tools. And they showed accuracy of their machine learning. So thank you. The third presenter, Nezenaya Onyechi. Please keep to time. Just keep your eyes on the clock. Once it's few minutes to please stop.
Good evening, everyone. Standing on all existing protocol, I'm Nizaya Oyenye Jacinta, a resident pharmacist of Chukwemeka Odumegwu Ojukwu University, and I'm here to present a research on evaluation of Nigerian plants as antifungal agents against malassezia for natural hair care product formulation. Like, so dandruff is a condition, is a scalp condition that is found in major countries of the world, but is mostly prevalent in men. Yes, men, not women. And then is characterized by flaky scalp, itchy scalp. But the thing is, there are actually products that have been made to combat these issues, but we face cases of resistance and side effects, which makes the need of a natural and safer alternative to be developed. So the objective of this research is to use computational methods to predict which plant compounds are best at inhibiting malassezia fungi, which is the fungi that is the primary cause of the condition dandruff. Secondly is to obtain antifungal extract using sourceless method from the chosen Nigerian plants that have these phytochemicals. Third is to confirm and quantify the antifungal power of the individual plant extract against the malassezia fungi or yeast in the laboratory that's found in the laboratory. Secondly is to develop a stable and suitable natural anti-dandruff shampoo incorporating these proven extracts. And finally is to verify the collective antifungal efficacy of the final shampoo formulation through controlled laboratory testing, that is antifungal assay. So the method, firstly, I did in silico method, which is molecular docking. I first carried out a literature review, analysis literature review to know the pathogenesis of this condition and then phytochemicals that have antifungal potential antifungal properties. I downloaded them from PubChem, the phytochemicals, and then the drugs, known drugs too, that have antifungal properties. Then also downloaded proteins from the fungi. And then the next thing I did was to convert them into the format for molecular docking, which is PDBQT, perform the molecular docking, post-docking analysis, and then when I was done, I got my frontliners and took it and found which plant contains or have like these phytochemicals. I got them and prepared them for metallonic extraction using saltless extraction method. Then when I was done, I got the extract, dried it to remove the excess solvent, and then carried out antifungal bioassay using the agar-well diffusion method of the 4 extracts, which are, the plants are Eucalyptus globulus, Aloe barbadensis miller, bitter leaf. The eucalyptus, for the Aloe barbadensis miller, I used the rind and the gel. Then I used this extract with the suitable concentration to form the shampoo and perform antifungal bioassay of the formulation. Next slide, please. This is the results for the molecular docking. So the criteria in which I used to make this table was these phytochemicals are phytochemicals that have proven to bind to cytochrome P450, the enzyme or hydrolase. Now, cytochrome P450 is 5-CZ1 is the fungi that is the protein that I use that is found in the fungi because of the structure, and this is also an enzyme that inhibits the conversion of sterol to ergosterol, which is important for maintaining the cell stability of the fungi. So from this table, we can see that these phytochemicals are potent. Next slide, please. Then the result, after this is the inhibition zone diameter that I got for various concentration. After performing serial dilution, you can see that eucalyptus has the highest inhibition zone diameter at 300 milligram per mil, and followed by aloe barbadensis and bitter leaf. Then this is the inhibition zone diameter of the product that I made. Next, and that's the image of the Petri dish. Thank you. This is the conclusion. So we found out that indeed crude plant extracts have antifungal activity, and then the formulation showed potency with mean inhibition diameter at 19.3 with standard deviation of 1.2 mm at 300 milligram per mil. Recommendation.
Thank you. Thank you.
So, just in conclusion, this is what the formulation looks like. It was made with a soap base of African black soap. Thank you.
Okay, thank you so much. In fact, when you read out your objective, I was actually waiting for that product. So thank you for showing us that product. Those are the things Professor Oloke, talked about. The first time I met him at Adeleke University, he brought his products to us. I said, these are the products that he has made. So I think that's very good. But you don't just manage your time very well, because we are really interested in the translational research that you did. So please go ahead and also ensure that you do animal studies to test the toxicity and all that. So thank you very much. The next presenter, Victor Olajide Akeshola. Please, presenters, once I say time, once you look at that time and it says time up, please just leave the podium, because we have 20, we still have like 15 presenters. Thank you.
Thank you. Standing on the existing order of protocol. Yes. This is a mathematical model for the progression and control of diabetes mellitus in African population. So, still talking about non-communicable diseases. Next slide. Yes. Mathematical models, they are just abstraction of reality. Models, so using mathematical concept, diabetes mellitus is a growing public health concern in Africa. So model help us to see how the mechanism and the interaction and as the disease progresses. So we did a simulation. Okay. It's all right. Move on. Next slide. So objective. We want to formulate a nonlinear mathematical model describing the glucose-insulin dynamics, then examine the effect of treatment, adherence, and lifestyle modification on disease progression. Then we did stability analysis, sensory analysis to see the control, how critical factors and how the interplay is controlled. Next slide. Okay, so yes, there you will see the other side, you see the mathematical model. It's a nonlinear system of equations capturing glucose and insulin dynamics. Of course, when you eat, the A that's the glucose intake that comes in when you eat and all that, and there is an outflow. And the second one, that's the insulin that is propped up so that your food is digested. So that's how the glucose-insulin dynamics, that's how it operates. Yes, we have done a lot of mathematics to come into this. Parameter estimation, Okay, now, yes, parameter, okay, next slide.
Please, let's have it quiet. Let's listen to them.
So now,
we can gain one or two things that we can also use
when
in
you
our
have mathematical models,
Please.
the first thing you want to do is possibility of solution. After possibility of solution, we did that. After that, we did equilibrium. So we had to do what happens when you do nothing, when the rate of change is 0. So, and there you will see that the model produces two stable equilibria. So we saw two equilibrium, is two equations, two equilibrium. So you have the stable and so two equilibrium, disease-free and the chronic state. Usually, if it is infectious disease, you will say it is endemic. But because this is a non-communicable disease, you won't say it is endemic, you say it is chronic because it continues in the system and it doesn't really go. So there is the disease-free, there is the chronic state. So those two things happens, okay. We did mathematical analysis. Sorry that I don't want to bore you with the mathematics. So there is the baseline. So if you look at the result, we have average blood glucose, insulin, that's then the sensitivity analysis, index, then the progression to chronic stage. So for the baseline we chose, of course, for a diabetic person, we use 9.5. That's the baseline. Somebody who has 9.5 milligram per mole of insulin is already a diabetic patient. So because for a normal person, it should be between 4.5 and 5.5. That should be the normal. So when you use the glucometer, you see its purpose and all that. So, but at 9.5, the person is already diabetic. So, but if with improved adherence, we saw that the blood glucose moved to 7.2. If lifestyle is also now included, if there is a lifestyle change, the blood sugar goes to 6.8. Though the person is still diabetic, you see that 6.8 is still a diabetic state. So, but with improved adherence, there is an improvement. Then the sensitivity index with 0.55 is the baseline. Now, with improved adherence, the insulin sensitivity index will increase. You see that. So without adherence, it will continue to go further. But if there is adherence to treatment, there will be an increased insulin sensitivity. Now, then, you look at the third one, the progression to chronic stage. There are two stages. It could be acute. There is complications of diabetes. You have complications. You have other comorbidity too. So now, with improved adherence, you see the progression to chronic stage is that chronic stage that many other complications continue, like neuropathy, nephropathy, and diabetic retinopathy, wound challenges, is that chronic stage that thing begin to increase. So if there is adherence, the progression to chronic stage goes to 38 with lifestyle change that is included into treatment. It goes to 29. So the model successfully captures the interventions. The mathematical model works. Data collection is still very good. So we want to encourage collaboration among mathematicians, clinicians, and public health experts. Thank you.
Thank you very much. So while the second presentation addressed the prediction, this fourth one look at the mathematical modeling of the chronic disease. So now we are now looking. I will call on the fifth presenter straight, please. Ajayi Oluwadunsin.
Good day, everyone. My name is Oluwadunsin Ajayi, and I'm representing my team on presenting this research, empowering African innovation in global health security through next-generation computational approaches for Rift Valley fever diagnostic biomarker discovery. Next slide, please. So Rift Valley fever, a zoonotic disease caused by a Rift Valley fever virus, was first discovered in Kenya in 1930 and has remained endemic across Africa for over 60 years, which is recently spreading to the Arabian Peninsula. Currently, the RVFV presents hemorrhagic fever in humans and causes devastating livestock losses with over 90% mortality rate in newborn animals. Currently, the diagnostic challenge is quite significant
as early detection is difficult because the disease presents no specific symptoms. And current methods such as ELISA and RT-PCR, they require advanced laboratory facilities which remain inaccessible to resource-limited settings. And with no human vaccines for RVFVF available, diagnosis remains critical. So this presents an opportunity for African computational innovation to bridge this diagnostic gap. Next slide, please. So our aim is to develop a novel multi-epitope biomarkers for rapid and accessible diagnosis of Rift Valley fever in resource-limited settings. And our objectives are to design chimeric biomarker constructs integrating B-cell and T-cell epitopes of RVFV, evaluating physicochemical properties of these biomarkers, validate biomarker protein binding through molecular docking with RVFV proteins to assess structural stability using molecular dynamic simulation, and to identify lead biomarker candidates for experimental translation. Next slide, please. So our computational workflow began with the retrieval of RVFV proteins through specific databases, and we subjected and then incorporating B-cell and T-cell epitopes for the design. Then we moved to 3 basic stages: the pre-construct stage to assess the epitope properties, then the construct stage, which has to do with the assembly of the biomarkers, then the post-construct stage for further validation. Next slide, please. So we designed our constructed RVFV biomarkers combines the design, like I have said earlier, the design of B-cell epitopes and T-cell epitopes, which are connected by short amino acid linkers to maintain the flexibility and the structure of the biomarker. Then our table 1 shows the physicochemical properties of the biomarker. Out of 4 biomarkers, just a particular biomarker, which is biomarker 2, had a strong and exceptional potential, which is why it is the only result represented here. And the figure 2 shows the well-folded structure of the biomarker, both in the 3D structure and when refined. Then the figure 3 shows us the solubility analysis of the biomarker 2. All of these results that I have here, they suggest that biomarker 2 is a safe, stable, and immunogenic diagnostic candidate for RVFV diagnosis. Next slide, please. So the molecular docking and validation. So biomarker 2 was docked with glycoprotein, which is the primary target for antibody recognition. And with these results, it showed this is the biomarker 2, this is the glycoprotein, and this is the docked complexes, showing the strong and specific binding. Then the protein-protein interaction of RVFV biomarker 2 shows the bonding between the biomarker 2 and the glycoprotein. Next slide, please. The molecular dynamic simulation of RVFV biomarker 2. This particular test was done to assess the stability and the behavior of biomarker 2 when docked with the glycoprotein. And all of these results represented here suggest that. So to my figure 8, the in silico cloning of the RVFV biomarker 2. This helps to confirm that the biomarker 2 is expression ready as it was successfully cloned into the PET28A+, and it shows that the biomarker 2 is actually expression ready for downstream experimental validation. Next slide. So our study outcome, we designed and validated 4 novel multi-epitope biomarkers with biomarker 2 having a strong and exceptional potential. And our computational framework enables rapid biomarker discovery and supports innovation across emerging diseases, providing foundation for rapid field deployable assays. And this is not just about a disease, it is about transforming our nation's diagnostic capabilities. Thank you so much.
Thank you very much. Thank you. So the team, they actually use immunoinformatics to be able to discover 4 biomarkers that could be used, as a form of to develop vaccine for this virus. So I believe that you collaborate with people so that you are going to try it in vivo and also to clinical trials so that we'll also be having your products in the market. Then the previous, that's the Akinshola Victor. Please, doctors in the house, try to collaborate with him, especially when you work on non-communicable diseases. So I think he needs collaboration, so you can see him after the presentation. So now we entertain 2 questions for the presenters. So just let us know the presenter your question is being directed to. So let's have it. Any question for the presenters? Any question? Any question? Any questions for them? No question? Okay. I want to ask the 4th presenter this question. That having predicted, I think your team, the other presenter could not come, you standing for him. So now, having predicted the communicable disease, especially the diabetes, I think, because you're talking of glucose and others. And your presentation also talk about the increase in all those parameters when intervention is not, applied. So, but when intervention applied, it goes down. And I want to ask that, what's the practicability of this prediction? How can it be used, in hospitals? That, or maybe even, is this something that can be used at home or something? What is the practicability? How can it be applied to us, especially in this locality?
Thank you. Well, the first thing is now that mathematics can be used for non-communicable diseases also. It's one thing we are establishing. Many times we usually talk about the SEIR model, that's the mathematical modeling, susceptible, exposed, recovered, and all of that. But in non-communicable diseases, there is no recovery. Everybody is susceptible. Not just age. You see that in the dependent variable we showed BP, we showed BMI, we showed age, we showed lifestyle. So he's saying now that, yes, your BP is very, very, very important. Now, it is not just age. You can have young people coming up with diabetes. So it is not age so much, especially in this generation. It's not age. It's much more on your BP, your BMI. So somebody can be young, but has a large and high value of BMI. So the person is a potential candidate for diabetes or hypertension. So that's one key thing there. And of course, what you eat is very, very important in the glucose insulin dynamics. So your food and adherence to treatment, then lifestyle. Adherence to treatment is very, 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 very important. Okay, so once it's set, what we have done is onset. It is already onset. You see that the baseline is for somebody who is already diabetic. That's why we chose 9.5. So we didn't look at pre-diabetic state. So, but once somebody is diabetic, treatment is very, very, very important. Adherence,
Thank you. Thank you so much. So please, you can. Please step down. Then.
All right, ladies and gentlemen, please a round of applause for. Okay, we still have one more.
No, I want to call
Okay, the next 6. Okay, no problem.
So I call on Samson A. Oni. You are here? Okay. Joshua Okpanike. Kolawole Temitope. Aladejano Oluwatoyin Modupe. Oyeniran Oluwatoyin Adeola. Please come up. Aladejana Oluwatoyin Modupe. Oyeniran Oluwatoyin Adeola. Lawak Waliyat. As the rule, you have 5 minutes. Once your time is up, please leave the podium. So I call on John Victoria. Samson A. Oni.
the chief host, the host of this event, our distinguished guest speakers, all professors present, my warm greetings to you all. My name is Samson Ayorinde Oni. I'm a 6th-year pharmacy student at the University of Ilorin, and the title of my presentation is A Deep Learning Approach to Uncover Novel Antimicrobial Peptides from the Human Microbiome. Next slide. The background of the study. Antimicrobial resistance is an escalating crisis and in public health, and it has been named by the World Health Organization as one of the top threats to global health. Not just that, even in our immediate environment, it is an issue. Once upon a time, an image was trending on the internet. It was an image of a antimicrobial susceptibility test that was snapped for it was for a patient in an hospital, and it was trending. What was there? It was showing resistance for different antibiotics. So a patient was resistant to up to about 10 antibiotics. So that's an issue, and it is a global threat according to the World Health Organization. So researchers, scientists, they are doing a lot to look for alternative to antibiotics that we have. Some are coming up with new antibiotics. But aside that, we also have what we call antimicrobial peptides. So antimicrobial peptides, they are like a new generation class of antibiotics. And this study explored antimicrobial peptides from the human microbiome. Next slide. The objective is to develop and validate a robust deep learning model to be able to predict antimicrobial peptides. And also, we apply that model to metagenomic data from human microbiome from different body sites. Next slide. So the method, what did we do? This study developed a robust deep learning model by leveraging transfer learning. So we used the ProteBERT algorithm, ProteBERT, that's protein bidirectional encoder for representation for transformers. So this algorithm is it was trained on hundreds of millions of protein sequences. So it is able to it works using NLP, which is natural language processing. It is able to classify or represent text as tokens, and it is able to predict the next text in a sequence. So it was the same thing that was applied to build this model, because antimicrobial peptides are all usually in form of text amino acid sequences. So we use the same thing for building this model. What did we do? We did a transfer learning. So using that model, we fine-tune it, and we're able to develop a model that can predict antimicrobial peptides. So after that, we now collected metagenomic data from different body sites, including the gut, the mouth, the skin, and the vagina, to computationally screen for antimicrobial candidates from that metagenomic data. We got over 40 million ORF, that's open reading frames, from the metagenomic data. Of these over 40 million open reading frames, after screening, we reduced it to 8 million plus. After that, we were able to reduce it to 5 million plus. And then we applied it to our model to predict antimicrobial peptides. The results, next slide. So after building our model, this is our model performance, and we did a comparative performance with other models. We used 4 popular models, which are class AMP, class AMP using SVM, class AMP using NNN, AMPIR, and DBAASP. These 4 models, they are popular models that are used currently to predict antimicrobial peptide. So the model we built, we compare it with these 4 popular models in use, and this was the result. We use accuracy, precision, recall, F1 score, and MCC, Matthews correlation coefficient, to as our evaluation metrics. And interestingly, our model had the highest score of 0.91, that's 91% accurate MCC. MCC is the evaluation metric that can be used as a kind of overall determinant. And eventually we got 13 top consensus AMP. That is, among the AMP we got, these 4 models also predicted them to be AMP. And next slide. So the conclusion, we're able to use our model to develop and 13 top AMPs. And the recommendation, those 13 AMPs, further study could explore in vitro and in vivo study to test them in the lab. Thank you.
Thank you for your presentation. So that team has actually used bioinformatics to be able to predict antimicrobial peptides in the era of antimicrobial resistance. So if you have any question for him, please pen it down. I call on John Victoria to make a presentation.
So good day to great minds in the house and to young scientists that are shaping the life of science. My name is John Victoria, and I'll be making a presentation on the topic Empowering Next Generation for Precision Medicine in Africa by harnessing big data, AI, and genomics for global biomedical innovation. Before I start, I would like to make an illustration. Imagine there is a production of a definite shoe size for everyone, not considering the shoe size, the leg size, and everyone have to fit into that shoe. How are we going to survive? Now, incorporating this into healthcare system, where there are discoveries, inventions in precision medicine, and we could find out that Africans have a low contribution into healthcare gene data in the biomedical state. And because of this, it has limited a lot of innovations to treat African-based diseases. And why did this happen? Because precision medicine is a kind of medicine that has to do with a study of a particular gene of people and population in order to treat diseases that affect them. Now, when there are limited supply of data of Africans alone in the biomedical setting, there will be a limited way African diseases can be treated. So here are the approaches in precision medicine. We need clinical genomics, that is the clinical gene makeup of an individual population, in order to know what is wrong with their genetic makeup. And in order to do that, we go down to their pharmacogenomics, which deals with how their gene, their individual gene respond into the diseases that is being occurred. And then the incorporation of AI intelligence. And with that, there is a proper healthcare system being made. Next slide. So the objective of this study is to map the current landscape of precision medicine in Africa, and also to review the progress that has already been taking place in precision medicine, and also to evaluate capacity building, how we can build capacity and make more innovations. And also we'll be identifying the gaps, challenges that are also in empowering the next gen. Next slide. So the method, we did a scoping review and searched through databases that are peer-reviewed journals, and also databases that are biomedical, of period of 2020 to 2025, with a thematic search of keywords on genomics, AI, and others. Next slide. So the result we found out is that precision medicine is already taking place in Africa, whereby we have innovators already making a space in precision medicine, the likes of H3A Africa, and also Nigerian 100,000 genomics project. What these people are doing in their own setup is to create genomic databases that are African-based in order to treat African-based diseases. And with their efforts, it makes us to tailor our research to only African-based diseases. And with that, we can create cure to African genomic setting using our gene makeup. With that, they find out that due to the variations in our genetic makeup as Africans, we have different makeup in how we respond to all these non-communicable diseases. And they were also able to find out variants of diseases that are only limited to African bases alone. And with that, they also, due to the research they made, they also find out how Africans also respond to some drug. Imagine using a malaria drug, the same drug, which worked for someone in the US, and you taking it here in Africa, it alters your body reaction. What has occurred? Just because of a change in location. that can be a cause. Just because of a change in your lifestyle, that can be a change, or how those things affect. Next slide. And here are the initiators, the country where they are based, they are focused in creating genomics database that are African-based. And here are those that, they are the achievements they've already made. Next slide. We could also see that incorporating electronic research record also will improve our healthcare system, the use of telemedicine, using of our internet. But one of the problems we find out is there are lack of infrastructures due to low light supply, internet connection being low, and other things like that. Next slide, please. So here are the challenges, and these are the opportunities that are being taken in place in order to improve our state in precision medicine. Next slide. Yeah, this is my conclusion on this, that precision medicine is here to stay in Africa, and if we can incorporate this, it will make our healthcare system better, and we'll be able to fight more disease. Next slide. So here are my recommendations. Thank you.
Thank you for your presentation. That's a good one. So, but I hope you will have enough data to be able to predict your precision, because it's a personalized one. You need so much data. So I hope you, as the work continues, you get more data. And these days too, that African too, they're also trying to have data privacy. So that one will also help us to have more data to be able to preside on what will happen to our population. Thank you so much. I want to thank you for this opportunity. I want to thank Dr. Kola Oladipo, Professor Luke. Thank you, sir, for this opportunity to anchor this abstract oral presentation session. I want to step down now, and I would like to call on Mrs. Ajay. Dr. Mrs. Ajay, please come up and take over from me. So let's give a round of applause for those that have presented. Those that are presented during the first session, let's give a round of applause to them.
Thank you, Ma. Good afternoon, everybody. Are you tired? You shouldn't be. We appreciate your patience since morning, and I'm sure you have learned a lot, and you have unlearned too. So we call on the next presenter. I don't have the list. Okay, please. Who is the next presenter? We are at number 7, right? As Olawale Temitope presented. Okay, please come up. You have 5 minutes, please don't forget.
Good afternoon, professors, distinguished guests, and my fellow researchers. My name is Kolawole Temitope. I'm from Redeemers University, and I'll be presenting on cancer-linked chromosome 1 variants show African ancestry associated frequency shift in 1,000 genome projects. These are my fellow researchers. Cancer, as we all know, is uncontrolled cell growth that is caused by genetic mutation in the DNA. Therefore, the risk is influenced by genetic variation, which differs between population. Like the just concluded speaker has said, these differences in population can influence how we respond to drugs, which cancer is more dominant in our midst in different populations. So most of these genetic cancer studies focus on European ancestry groups, and it leaves African population underrepresented. According to a report, Africa contributes to a mere of 0.01% of the global peer-reviewed research in majority of journals. And this imbalance can lead to misinterpretation of genetic variants that are important in our population. So what our study do is to ask whether certain variants at different frequency can affect, or if they are significant and can affect how we respond to cancer in our population. So our study focused on chromosome 1, which has been previously linked to have several areas that are genetically mutated and linked to cancer. Next slide. So, next slide. So our study aimed to identify ancestry-related variation in cancer-associated regions in chromosome 1. And we also aim to provide a practical approach for examining such pipeline using public data sets. Next slide. So we use the Human Genome Project, the 1000 Human Genome Project, and that's on chromosome 1 specifically, and the analysis for Human Genome Project contains about 2,504 individuals across 5 superpopulations, which include Africa, America, East Asian, European, and South Asian. So what we did is we downloaded the chromosome 1 data set, including the sample panel, which is the metadata, and we isolated the regions according to papers, the regions that are linked to cancer in that 8 specific loci that are linked to cancer. So we extracted the variants using the VCF file particularly, and we keep the single nucleotide variants using the plic set. Then we compute the allelic frequency for those 5 frequencies, and we plot it by population. And then we did functional annotation for that variant to check which of those variants are functionally important. Next slide. Next slide. This is the flow chart showing the method we use. So our results, as you can see, the African population and the other population cluster, that is the variants that are linked, on chromosome 1, the variants that are linked to cancer clustered in other 4 populations. So the PCA shows that African population clearly separate from other populations, and they don't just clearly separate, they are genetically diverse, they are very diverse, which shows that each and every one of most African population have distinct variants that are linked to cancer. So non-African groups cluster closely together, which shows similarities, and that confirms that there is a distinct genetic structure in those regions. That's the heat map showing the variants. Next slide. So we also did the allele frequency, and we compare it with each of the alternate allele or heterozygotes and the reference. Amidst those, it is the African population that have the most variance when we compare it with the reference sequence. And that supports that African population is genetically diverse, as we have said earlier, and the American shows the least. Next slide. Out of those variants, we extracted the African population variant. Then the total variant we extracted was 264, and we also conducted the false discovery ratio, and we extracted the significant variant, because it is not all of those variants that are statistically significant, and out of those, it was 27 of them that were statistically significant at FDR, false discovery ratios less than 0.0. That's 10.2%. Previous slide, please. So 3 of those variants, no, previous. Okay, yes. So 3 of those variants, we conducted functional annotation.
Your time is up.
Okay, 3 of those variants were significant, and they have high functional annotation, and they were of high impact. That is, they have ability to conduct eye disease in African disease, but there are no African private variants that we discover. Next slide.
Please,
Next
your time
slide.
is up. Hello.
Thank you.
Yeah, that's the right thing to do. Thank you. We won't be making comments again for now because of our time. Please, is Goodness Akorede Akapo here? Is he in the front? Please, you can come if you're okay. I'll call on Joshua Opanike. Oh, sorry. Opanike.
Hello.
Go after him.
Distinguished professors, erudite academics, distinguished ladies and gentlemen, my name is Joshua Opanike. I'll be presenting a case for improved genomic surveillance of mpox and evolutionary and phylogenetic analysis. Next slide. Mpox is a zoonotic viral disease. It's caused by monkeypox virus, a double-stranded DNA virus of the poxviridae family, and recently emerged as a public health issue when it escaped its usual geographic boundary, usually in Central Africa. Clade 2B was one that was associated with the worldwide spread that caused a lot of scare in 2022 to 2024. And it found that there was rapid diversification in that group and has led to sustained human-to-human transmission outside of Africa. They also found that the virus has an adaptive reference to new epidemiological settings, and this has led to emergence of novel sublineages, and this warrants continuous genomic surveillance. Next slide. And so our objectives were to contribute to the genomic surveillance of Mpox in West Africa. We wanted to investigate the genomic diversity of Mpox in the region and to also identify evolutionary patterns of Mpox in the region. And so our methods, we retrieved sequences from 2019 to the 24th of August of 2024 from the GSA database. The data sets were organized according to country of origin, clades, and year of submission, and we used multiple alignment fast Fourier version 7 web server to determine common ancestors, and we also did comparative analysis using the same tool. Next slide. And so results, we found out that in West Africa, there were only 4 countries that had genomes, that had sequences, and only 2 of them, Cameroon and Nigeria, had complete sequences. And if you can see on the other side, the graph is showing that actually during that time frame, the sequences that were deposited on GSAID were in the clade 2B. Next slide. And so our results, we found that all isolates shared consensus similarity with the reference sequence, indicating that there's a common ancestor and that this spread was from a similar origin. And we also found that the sequences have 98.5% identified CAR sites and 99.5% pairwise identity. Also means that despite the fact that there is so much diversity, which the MSA also showed, with many polymorphisms, still the lineages are similar in a way. Next slide. And so that's what we found, and our conclusions is that our findings align with recent studies that show increasing genomic divergence within clade 2B, and that the predominance of clade 2B sequences in our data sets reflects that there's an ongoing global spread and the microevolution of monkeypox virus, particularly following that previous slide. And we also found that there is sustained human-to-human transmission of monkeypox virus within West Africa. Next slide. And so we recommend that there should be more investment in genomic infrastructure. Like I mentioned, we had data from just 4 countries. That is not, and then the ones that we had, a number of them were not quality sequences. We also recommend that we should continue sustained genomic surveillance of Mpox. Despite the fact that most of these sequences seem to be related, we should also continue the work that we are doing, especially the vaccination effort that is going on in some parts of northwest Nigeria. And the role of animal reservoirs in Mpox evolution should be studied. In our study, we only found sequences from humans. We couldn't get sequences from animals. And this is very important, especially to ensure concerted efforts in a One Health approach. Next slide. We'd like to acknowledge the efforts of Global Initiative on sharing all influenza data for hosting the sequences on the server. So I want to appreciate the Director of Science and members of staff and interns at Elix Belgian Institute, as well as acknowledge other co-authors, including Dr. Oladipo, Mr. Stephen Adem, who was a consultant, Mr. James Oguniro, Ms. Possible Popola, and Ms. Victoria Alabi. Thank you very much.
Thank you for managing your time well. I'll call on Goodness Akorede.
Good evening, everyone. My name is Goodness Akorede Akakpo, and I'm here to present on a recent research done by I and my research group titled A Novel Multi-Epitope Subunit Vaccine targeting the VP6 protein of rotavirus. Next slide, please. Rotavirus is a leading cause of severe gastroenteritis in children less than 5 years, and it's responsible for 128,000 deaths annually. Existing vaccines such as Rotavac, Rotacil show very low coverage, most especially in regions such as sub-Saharan Africa. Next slide, please. Our objective was to design a novel multi-epitope vaccine with a broad coverage against diverse rotavirus strains. Our other objective included to design a vaccine that could induce CD8, CD4, B cells, and could also have, is also non-toxigenic and non-allergenic. Next slide, please. So the entire process started from sequence retrieval all the way to multiple sequence alignment, then to epitope prediction from B cell epitopes, CTL epitopes, then HCL epitopes. Next slide, please. Then we went on to carry out an epitope assessment, then on to population coverage, vaccine construct design, then physiochemical property analysis, then prediction and visualization. Next slide, please. Then we went on to molecular dynamics, immune simulation, in silico solubility, before we ended it all with in silico cloning. Next slide. Next slide, please. So after epitope prediction, we landed with 9 B cell epitopes, 5 CTL epitopes, and 5 HTL epitopes. The selection criteria included safety, high conservancy, antigenicity score, the cytokines they induce, and the peptide length. And as you can see from the table, we can see the criteria that was used to select. Next slide, please. Next slide. So from the population data, you can see the various population coverage of these individual epitopes. And if you notice, you can see that CTL epitopes has a very broad coverage, mostly and most importantly across Africa. Next slide, please. And you can see an horizontal bar chart showing the difference in the percentage coverage of these individual epitopes. Next slide, please. Next slide. So the vaccine construct was joined together using various linkers such as KK, AAY, and GPGPG. We made use of 3 CTL, 3 B cells, and 3 HTL epitopes to construct this vaccine. Then we made use of FLIC-C as our adjuvant due to its ability to induce both cell-mediated immunity and, yes. Then for the vaccine construct 13, we carried out a physiochemical property analysis to check the instability index, the molecular weight, aliphatic index, and also the GRAVI. And it indicated that it was a stable vaccine. It was also thermostable and nitrophilic. Next slide, please. Next slide. Then we carried out an in silico solubility and fusion tag analysis to see which fusion tag will be best for purification later on. Next slide, please. Then we went on to tertiary structural prediction, then also refinement and validation. And from the Ramachandran's plot, you could basically tell that 97% of the residues were in the favorable region, while just 3% of the residues were in the unfavorable region. The ProSA-Web Z-score also gave us a score of minus 9.82%, confirming that it is a structurally stable protein. Next slide, please. So you can see the Ramachandran's plot here. We can also see the energy score plot indicating the negative ProSA-Web Z-score. Next slide, please. Then we went on to docking, then MD simulation, which told us that we had a rigid and stable construct. We also had a very flexible construct. Then it was also the solvent acceptability surface area result also indicated that the protein was dynamic, but it is a compact structure. Next slide, please. So you can see the RMSD data. Next slide. You can also see the RMSF, radius of gyration, the solvent accessibility, and the number of hydrogen bonds. Next slide, please. Now, for the immune simulation, our construct shows that it could induce key cytokines such as interleukin-2, interleukin-4, IL-10, tumor necrosis factor, alpha. Conclusion, please go to the end. Go to the last slide, please. Last slide, the acknowledgement. Special thanks to Professor B.O. Alokun for the opportunity, guidance, and mentorship. Also, special thanks to the organizers of this conference and finally, special thanks to God Almighty. Thank you very much.
Thank you. I call on the next presenter. Please put it there. Oluwatoyin Mojupe Ade Aladejama. Oluwatoyin Mojupe Aladejama. Oh, okay. She's not here. Oyeniran Oluwatoyin Adeola.
Good afternoon, everyone. I'm here to present the topic, high prevalence of antigenic markers and genetic variability of Plasmodium falciparum in Osun State, Nigeria. My name is Oyeniran Oluwatoyin Adeola. Next slide. Malaria remains a public health issue in Nigeria, contributing significantly to morbidity and mortality rates caused by Plasmodium falciparum, and this parasite exhibits profound genetic diversity due to high rates of meiotic recombination during sexual reproduction, which allow rapid adaptation and immune evasion. These service markers are crucial for parasite survival and immune responses modulation, making them central target for potential malaria vaccine. Co-infection with HIV alter malaria pathogenesis and may influence transmission dynamics and selective pressure of parasite population in endemic regions. Next slide. Next slide. Objectives to determine the overall prevalence rate of Plasmodium falciparum infection among individuals in selected areas of Osun State, to assess frequency, distribution, and prevalence of key antigenic markers across the sampled parasite isolates, to determine and compare genetic variability within P. falciparum isolates, and to also assess and map phylogenetic and evolutionary relationship among isolates collected from 3 major sampling sites. Next slide. Materials and method. A total number of 422 venous blood samples were collected in consenting participants. An ethical approval was also obtained and informed consent secured from all participants, and the subjects were classified into cohorts based on HIV status and clinical presentation, and all samples were subjected to malaria microscopy for detection, and also the antigenic marker was confirmed by using nested PCR. Next slide. The genomic analysis pipeline. The genomic analysis was performed using standardized workflow in integrating sequence annotation and advanced bioinformatic tools. Whole genome sequencing utilizes Illumina ISEQ 2500 system to generate high-quality sequence for selected isolate. Sequence were annotated using companion server and mapped against reference genome via NCBI BLAST, and genomic relationship and molecular evolutionary analysis were computed using Mega version 5.2 software package. Next slide. The result. Overall prevalence rate of 22.9% were confirmed by microscopy, and the antigenic marker was striking. High prevalence rate indicates significant exposure and circulation of diverse parasite genotype. The MSP1 and MSP2 were found in a massive 96% isolate, and this indicates, it highlights the role in human immune invasion, and the genotype, the gametocyte was found to be 90% in the isolates analyzed, which indicates a higher potential for transmission within the population, and this marker is associated with sexual stage of the parasite. And while we also found a moderate presence of glutamic-rich protein, which is part of the antigenic surface markers of the parasite. Next slide. The sequence length, there was a significant difference was observed in the length of the sequence isolate, indicating complex polymorphism, especially in MSP1 and MSP2. Isolate demonstrated variability in their GC and AT content, suggest distinct genetic backgrounds and potentially different evolutionary pressure across the three sites. Higher diversity in the viral gene composition was observed, which is critical for cytoadherence and virulence, reflecting robust immune selection. Conclusion. P. falciparum population in Osun State exhibit extensive antigenic diversity and higher genetic variability, which shows that there should be a high robust vector control and chemoprophylaxis. Next slide. Recommendation. Continuous monitoring is essential for molecular surveillance.
Recommendation. Let's have the next presenter, Lawal Waliat, and she'll be the last presenter for this round. Waliat. Lawal, right?
Yes, ma'am. Good day, everyone. My name is Lawal Waliat Ayomide, and I am here on behalf of my team to present our work on targeting antimicrobial resistant gene in Acinetobacter baumannii using in silico technique through antimicrobial peptide. Next slide, please. Acinetobacter baumannii has become a global threat in hospital as the organism is becoming more resistant to antibiotics such as carbapenem. This organism is and this organism is a multi-drug resistant organism that is associated with hospital-acquired infection and ventilator-associated infections. Next slide, please. This is a global epidemiological statistic that is showing the prevalence of Acinetobacter baumannii in Europe and USA, and the other slide showing for the HPI and the HAI. Next slide, please. The objective of our study. Our first objective is to identify and select plant-based antimicrobial peptide with predicted antimicrobial activities. Our second objective is to evaluate the interaction between the antimicrobial peptide and the Acinetobacter baumannii resistant gene. Our third objective is to identify the ability of our selected antimicrobial peptide in targeting a baumannii resistant mechanism. Next slide, please. Method that was used. First, we retrieve our data from the DRAM database, which was filtered to the plant source. Next, their allergenicity, hemolytic, and toxicity analysis were predicted, followed by the physicochemical characterization. Next, we went through literature databases to search for literatures, and we were able to find key baumannii resistant genes that were retrieved from the UniProt database. The 3D structure of the antimicrobial of the AMP, which is the peptide, and the antimicrobial resistant genes that were identified were predicted using AlphaFold. They were docked with a HDoc, and a PDB sum was used to actually identify the key proteins that were present in the interaction. WebGrow was used to carry out the molecular dynamics simulation, and the ADMET predictor was used to identify the absorption, metabolism, excretion, and distribution of this of our peptide. Next slide, please. Okay, so after filtering, these are the two peptides that passed the criteria that was set, which is the Cucurbita maxima and then the Arabidopsis thaliana. Next slide, please. For our results, these are the 3 key genes that were used for this analysis, which is the Acinetobacter drug F-B, which is ADB, PMRB, and AMPC gene. And these are the mechanisms which they use in working, and they are antibiotics that they are resistant to. Next slide, please. Okay, so this is the molecular docking results. The first figure here shows that of the AMP in red, the gene in red. This gene is the only gene that the organism, the peptide that were gotten, were able to bind very well to. Next slide, please. So these are the docking results, and our docking results here shows during the analysis, we actually got an approved peptide that was used as a positive control for our analysis, and which is gramicidin. So these are the this is the ADEB gene, which Cucurbita maxima, and our results here shows that our organism, our peptide bind more to the ADEB gene than gramicidin that was already in the market. Next slide, please. So this is the ADMET results that showing the distribution, and these are results. The first one here shows for gramicidin, and the other one shows for our peptide. This result simply means that our organism have the ability or the potential to stay more in the infection site and battle the organism, and also have the ability to, it won't cause any side effects to the central nervous system. Next slide. This means our organism is soluble. Next slide. And this shows that our organism have no side effects on the liver, kidney, and even will not cause any skin infection when in comparison to gramicidin. Next slide, please. Conclusion. So our study on plant-derived antimicrobial peptide actually shows a strong potential in battling BI0MEDICAL-resistant genes. Next slide, please. Recommendation. We recommend that in vitro and in vivo analysis should be carried out on this predicted therapeutic peptide, and optimization for the stability and bioavailability should be done for better drug performance. Molecular docking and dynamics should also be carried out for better peptide and target interaction. Next slide, please. We acknowledge Ellis Biology Institute for giving us the infrastructure.
Thank you, Aliat. Your time is up. Yes, we thank all our presenters here. You can go back to your seats. Thank you for contributing your quotas to scientific body of knowledge. I'll call the next 6 presenters. We are almost getting to the end of the section. Shola Peter Ogumodede. Shola Peter Ogumodede, are you there? Please come up. Olakomi Oluwakemi Deborah. Elutade Olubumi Ayobami. God save Binlak Wahiz. I don't know if I correct that, if I pronounce that correctly. God's will Antonin. Let me go over it again. Shola Peter Ogumodede. Olakomi Oluwakemi Deborah. Elutade Olubumi Ayobami. God save Wazi. God's will Antonin. Iwachukwu David. Mercy Bisola Fayemi. Faleyemi. Faleyimi. Okay, Emma Binu. Faleyimi. Okay. Erioluwa Tinuola Taiwo. Okay. Now, I'll call on Shola Peter Ogumodede to come to the pulpit. Okay, he's not available? All right. Olakomi Oluwakemi Deborah, you have 5 minutes, please don't forget. The moment I say your time is up, please, your time is actually up.
Good day, everyone. Good day, everyone. I am Olakomi Oluwakemi Deborah. I'm here to present on a topic which says effect of vitamin D3 on renal lipid handling in crystal methamphetamine exposed rat. Next slide, please. Methamphetamine, popularly called MELT, is a powerful stimulant drug with high abuse potential. And methamphetamine is a colorless, tasteless, and bitter-tasting drug which can easily dissolve in water. Chronic crystal melt exposure causes renal damage through oxidative stress and lipid imbalance. Chronic crystal melt majorly affects the central nervous system, which is the brain. Its high exposure affects the renal system, which causes the oxidative stress and lipid imbalance. Vitamin D3 is one of the fat-soluble vitamin, and majorly it is gotten from the sun exposed. This study aims to evaluate the effect of vitamin D3 on renal lipid handling in crystal methamphetamine exposed rat. Next slide, please. Objective. The study of this is to evaluate the influence of vitamin D3 on renal lipid and crystal methamphetamine exposed rat by assessing the morphological changes, which is the body changes and the relative weight changes, measuring renal function diseases, which is the pyruvate assay, the renal assay, evaluating renal lipid profile on the kidney imogene, which is the triglyceride, lower density lipoprotein, high density lipoprotein, and the histological changes of the kidney. The next slide, please. The method: 24 male Wistar rats. They were the 24 male Wistar rats were randomized and they were grouped into sections. They were grouped into four groups, and there were 6 cages inside a rat. The first cages was labeled the control, which was not given anything. The second was methamphetamine. The third was vitamin D3, and the fourth was met plus vitamin D3, and they were administered, the drug were administered orally using oral cannula for 4 good weeks. The next slide, please. And the results when we measure the morphological changes, the result here shows that the body weight, when compared to the vehicle group, which is the control group, it reduced the body weight. While the relative renal changes, when compared to the vehicle group, which is the control group that was not administered anything, it increases, which makes vitamin D3 to suppress the methamphetamine. The next slide, please. The results for the serum creatinine and the serum urea, which means methamphetamine in they both increase. They both increase when compared to the vehicle group, which is the control group that was not administered anything. The next slide, please. This is talking about the renal lipid profile, which is the high density lipoprotein and the low density lipoprotein. In high density lipoprotein, the methamphetamine was drastically reduced when compared to the vehicle group, which was not administered anything, which is the control group also. While the low density lipoprotein, the methamphetamine was increased, which causes the vitamin D3 to suppress the both. The next result, please. This is talking about the histological changes of the body, where we can get the signs of the injuries. Here is talking about the methane in the histology of the body drastically reduce it, which causes renal degradation, and the vitamin D3 causes the lesion. Can we go to the next slide, please? Conclusion: this study demonstrated that vitamin D3, when taken to suppress methamphetamine, it actually reduces it. And also the reaction in histological damages further supports vitamin D3 in preserving renal tissues integrity. The findings align with the research on vitamin D3 and anti-inflammatory and anti-hypotonic properties, making it a promising agent for integrating metal-induced renal toxicity. The next slide, please. Recommendation: research should be conducted on a well-developed structured study design in both in vivo and in vitro experiments. Also selecting appropriate biomarkers for the assessing the renal lipid handling. Also techniques such as the quality PCR should also be used. The next slide, please. I give thanks to God Almighty and also to my supervisor, which is here, Prof. A.F. Ajayi. I say thank you to you, sir, for your guidance, support, and valuable contribution. Also, thanks to the committee of this organization and to everyone who contributed to the. I say thank you. Thank you for listening.
Thank you, Ms. Deborah, for managing your time well. I call on the next person, Elutade Olubunmi Ayobami.
Good evening, everyone. My name is Elutade Olubunmi Ayobami, and I am representing my group to present on a project titled Utilizing Immunoinformatics for mRNA Vaccine Design Against Influenza D virus. All right, so influenza D virus is a single-stranded RNA virus that was first discovered in 2011 in a pig. It has 7 proteins, some of which are the nucleoprotein, the matrix proteins, and the hemagglutinin esterase fusion protein, which is the most immunogenic protein because it is found in the outermost surface layer of the virus. Although there are no reports of any human infections yet, the possibility of human susceptibility to the infection cannot be ruled out. Hence, the need to apply immunoinformatic methods to create an mRNA vaccine against IDV. Next slide, please. So the objective to design a universal mRNA vaccine against influenza D virus. The methods: we retrieved IDV, HEF we retrieved HEF sequences from NCBI. Then we mapped out the CTL, the HTL, and B cell epitope. And then we moved to the construction of the vaccine. The final construct was then used to generate the secondary and the tertiary construct. The tertiary construct was refined, validated, and subjected to other post-construction analysis, such as the molecular docking results. 88 HEF sequences were retrieved across all continents. HCl, CTL, and linear B-cell epitopes were predicted and analyzed. We move on to the construction stage. So 11 final epitopes were selected based on their high antigenicity and immune response potential. Additional components such as the 5' UTR, the Cossack sequence, the TPA were incorporated into the primary construct. Then the primary construct was predicted to be antigenic, non-toxic, and non-allergenic. So to the post-construction stage, the primary construct was used to generate the secondary construct, which can be seen in figure 6, and then used also as well to generate the tertiary structure, which was then validated, and this is the result for the validation. Next slide. So the tertiary construct was refined and then docked with TLR2 and TLR4. And TLR2 showed lower binding affinity compared to TLR4. This signifies the potential for triggering an immunogenic response. Next slide, please. So due to the significant threat posed by the IDV, this study leveraged on immunoinformatic approaches to design an mRNA vaccine design. However, further in vitro and in vivo studies are required to validate the safety and efficacy of this vaccine candidate. Next slide. So for recommendations, further post-construction analysis such as MD simulation, in silico cloning are required to enhance this study, and collaboration should also be made to strengthen between veterinary and public health agencies to improve monitoring, diagnostics, and research on IDV transmission. Next slide. So we, all the authors, gratefully acknowledge the support of Helix Biogene Institute for providing this enabling research environment and resources required for this study. We also accord special thanks to collaborators, institutions who also publish data and tools which form the basis of our immunoinformatics analysis. Our sincere appreciation goes to every person that participated and contributed to this project. Thank you.
Thank you, Ayobami. We are almost getting to the end of this presentation. I call on God Save Binlak, right? What? Wazi.
Waji.
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 Oh, I see.
Good evening, everyone. My name is Godsave Binlak Waji, and I'll be presenting on the epidemiology and comparative evaluation of diagnostic methods for hepatitis C virus detection among adults attending primary health care centers in Jos Plateau State, Nigeria. Next slide. These are my co-authors and their affiliations. Next slide. So the hepatitis C virus is still a global health challenge. The statistics are staggering. 58 million people live with the chronic infection, and about 1.5 million new cases are detected annually. So many stakeholders are shifting focus away from hepatitis C to other infectious disease, let's say hepatitis B. Meanwhile, while B has a vaccine, C does not have a vaccine, and even though it is treatable, it is known as a silent killer because many people remain asymptomatic until late on in its course. So many regional disparities still exist in Nigeria, and that is why it is important for reliable diagnostic strategies and early detection for this virus. Next. So these are my objectives: to determine its seroprevalence, to determine the association between some risk factors and the infection, to evaluate diagnostic strategies, and then to confirm the RDT and or ELISA positive samples using the reverse transcriptive qPCR. Next. So this is my method section. It was a cross-sectional study done in JOS. So in JOS, there are 3 local governments. Each of these local governments is a cluster. And this calculated sample size is 270 adults. These 270 adults were distributed amongst the 3 PSCs based on a proportion to size allocation formula as adapted from the study by Taufik. So the PSCs were selected based on a simple random sampling by balloting. As you can see, PSC Tudunwada from Jos North, PSC Bukuru Express from Jos South, and PSC Fobor A from Jos East. Structured questionnaires were used for demographics and risk factors assessment. For the serology, we used the RDT and then a 4th generation ELISA which detects IgM and IgG. Next slide. And then for the molecular confirmation, samples which were positive by RDT and or ELISA were tested using the Lunar Universal One-Step Reverse Transcriptive qPCR kit. Positives were determined based on the cycle threshold plus a single mel point. So these were the formulas used to calculate the performance parameters. Next slide. For the results, the seroprevalence by RDT was 7.0% and then 7.8 by ELISA. The reverse transcriptive qPCR confirmed 18 out of the 21 samples which were positive for the RDT and all the ELISA positives. If you look there, you see that 19 samples were positive by RDT, 21 by ELISA. All of the samples that tested positive by RDT also tested positive by ELISA, but 2 samples that tested positive only for ELISA. So if you look at the intersection of the Venn diagram there, out of the 19 that tested for both of them, 17 were PCR positive, 2 were negative, and then the 2 samples that tested positive for only ELISA, 1 was positive, the other one was negative. For the risk factors, the only significant ones were the history of STD, a spouse having hepatitis C virus, and then previous history of STD indicating repeated exposure risks, and then illicit injection of drug use, previous blood transfusion, history of surgery, and other risk factors were not significant. Next. So at the end of the study, when you use the ELISA as the serological gold standard to compare the RDT, it was 90.48% sensitive and then 100% specific. The accuracy, which is the efficiency, was 99.26, and then the positive predictive value was 100. Next slide. So the conclusion is that HTV remains a notable health challenge, although the ELISA was slightly higher in sensitivity, but the RDT still remains a reliable tool, especially in low resource settings. But then the PCR should always be, the PCR should always be done to confirm active positives. Next slide. The recommendation is that since there's a strong concordance between the RDT ELISA and RT-PCR, we can use the RDT as a rapid alternative in low resource settings, while also trying to strengthen surveillance systems and promote genotyping. Thank you.
Thank you, Mr. Wai. You can drop it there. I'll call on the next. It seems God's will, Anthony is not available. Okay, Nwachuku David. We have just 2 more to go. Just 2, yeah. Just those 2. Have the floor.
Good evening, all protocol. Assemble. My name is Nwachuku David, and I'm here to present the topic of seroepidemiological characterization of dengue IgG antibodies among HIV positive inhabitants in 2 tertiary hospitals in Anambra State, Nigeria. A brief background: dengue virus causes dengue fever, which is spread mostly by the Aedes mosquito, especially the Aedes aegypti mosquito. It can also cause severe dengue, which can be dengue hemorrhagic fever or dengue shock syndrome. Now, the objectives of this entire work was to find the past infection seroprevalence rate among the HIV positive inhabitants in Anambra State in these 2 tertiary hospitals. It was also to find any socio-demographic or clinical risk factors that could promote the distribution of this disease. as well as any significant relationship they have. Now, the materials and methods, the two hospitals looked at were the St. Charles Borromeo, as well as the Chukwuemeka Odumegu Ojukwu Teaching Hospital, Kowut and Borromeo for short, respectively. 94 participants came from each, giving a total of 188 participants. This was a cross-sectional study done with specific inclusion and exclusion criteria. Now, this had three steps. The first was the serological tests. The second was the data analysis, where we grouped them within the socio-demographic factors, and the final was the statistical analysis to see if there was any relationship. Now, on to the results. The first result shows us the overall seroprevalence, where it gave us a 78.2% overall. Kowut was high with over 90%, whereas Borromeo had over 60% seropositivity of this virus, of IgG dengue. The next result shows us the social demographic and clinical characteristics, where those aged 61 to 75, females, married, those with no formal education, those with a viral load of over 1,000 copies per mil, and those with a CD4 cell count of less than 200 cells per microliter had the highest IgG seropositivity in each of their groups. Now, in conclusion, there was a high IgG seropositivity across all groups and all risk factors. However, marital status and educational status, based on our statistical analysis, were the only ones showing a significant factors that is under a 95% confidence interval. Now, my recommendations, all the 4 recommendations there based on are simply telling us, first, those with HIV need to know about this disease because they are immunocompromised. While this disease may cause asymptomatic or mild symptoms in a normal person, it can cause severe dengue on them, and that can be life-threatening. Also, promoting routine dengue virus tests on HIV participants whenever they come for checkups, as well as telling them how to prevent mosquito bites and how to prevent this disease. Finally, I wish to acknowledge Pamo University of Medical Sciences, especially my coordinator, Professor Boysa. I wish to acknowledge those who worked on this with me in the University of Port Harcourt, especially my supervisor, Professor I.O. Okonko. Finally, I wish to thank the Heligen Biogen for giving me the opportunity to present my work here today. Thank you very much.
Thank you, Mr. David. He was so familiar with his slides that he did not even check anything. Did you notice that? That is a good presentation skill. He spoke to the slide. He wasn't reading at all. Thank you, Mr. David. The next presenter, Mercy Bisola Fale Yimu.
Good day, everyone. I am Mercy Bisola Fale Yimu, and on behalf of my group, my team member, I am here today to share our findings on the study Bridging Barriers to Accessing Clinical Trials and Advanced Therapies for Liver Cancer in Sub-Saharan Africa, a systematic review from the year 2005 to 2025. Liver cancer, especially hepatocellular carcinoma, is a very serious cancer. It's one of the most serious cancer worldwide, and it is driven by factors such as hepatitis, infection, aflatoxin exposure, and alcohol use. It is one of the major health challenges that people in sub-Saharan Africa face. And clinical trial it plays an essential role in helping to develop advanced therapy for liver cancer. Moving forward, the aim and objective of this study is to assess the availability and the accessibility of clinical trials in sub-Saharan Africa, to assess the factors that help or hinders patient enrollment into clinical trials, and lastly, to develop evidence-based policy so as to be able to make improvements in the development of treatment. Method. For our method, we searched 6 major databases and were able to retrieve 111 articles. After our screening and eligibility assessment, only 16 studies passed our review. And can we move forward, please? Next slide. So showing from my first result, from the 16 studies that were reviewed, only 11 countries in sub-Saharan Africa were represented, mostly in Ethiopia and South Africa. This shows that there is a need for more studies on liver cancer in sub-Saharan Africa. From the second result, we can see that 56% of the studies shows that it does not specifically explain the type of access received, and 25% of the studies shows that there is limited access, while 19% of the studies shows that there is no access to clinical trials in sub-Saharan Africa. Moving forward, we can see here that the treatment access types, only 11% of patients received curative advanced treatment, while 22% of the patients received curative treatments, and lastly, 31% of these patients received palliative treatment. This shows that treatment outcome in sub-Saharan Africa for liver cancer is very low due to late diagnosis and poor health infrastructure. Moving on, in conclusion, late presentation, lack of financial resources, insufficient trained personnel, and inadequate healthcare infrastructures are the major barriers to accessing clinical trials in sub-Saharan Africa. And to address this issue, there is a need for intervention targeted towards our healthcare system so as to be able to help patients living with liver cancer. Moving forward to my recommendation, to improve better health outcome, there is a need for implementation of HPV vaccination, establishment of screening, treatment, and surveillance program for HCCC. There is need for public health awareness so that people can know that, yes, this and this are what can cause HCC. The use of validated biomarker for early diagnosis, and lastly, we should foster clinical and genetic research collaboration based on African population. My acknowledgement. We, the authors, would love to appreciate Helix Biology Institute for this great platform to present our work, and to our supervisors, we say a big thank you, and to all the databases that we retrieve data from, we say thank you.
And to Bisola, we say a big thank you. Your time is up. I would like to call the last presenter for this presentation round. I think this is the last one. There is no other one. Don't bother. Erioluwatinu Olataiwo.
Good evening, everyone. All protocols duly observed. I'm Erioluwatinu Olataiwo. I'll be presenting a microbiological assessment of bacterial contaminants on canned drink surfaces sold in selected areas in Ogbomoso. Next slide. Most of our canned drinks, we are familiar with canned drinks because of its convenience and affordability in many countries of the world. And many microorganisms survive on the surfaces of these canned drinks. And then ingestion of these microorganisms can lead to diarrhea, food poisoning, among others. Next slide. The objective of this study is to identify and to detect possible pathogens from canned drink surfaces, to assess the public health implications, especially antibiotic resistance of the bacteria isolate. Method, the samples were collected, alcoholic and non-alcoholic samples were collected, and there were 3 of them, of each sample were collected. One of the surfaces was disinfected with 70% alcohol, and then one was rinsed with running water, and the other was left neither disinfected nor rinsed. Then the bacteria was isolated via swabbing and streaking technique. Gram staining reaction was carried out. Biochemical tests were done, oxidase and catalase tests biofilm formation was done, hemolysis testing, as well as antibiotic susceptibility testing via the disk diffusion method, also known as the Kabaiba method. Next slide. Our result shows that more bacterial isolates were gotten from surfaces of non-disinfected canned drinks as against rinsed or disinfected canned drinks. It also shows that more isolates were observed on nutrient agar because it is a non-differential and non-selective agar. The result shows that, previous slide, the result shows that more bacterial isolates were gotten from surfaces of alcoholic canned drinks, and this poses threat to consumers of alcoholic canned drinks. Coccobacillus was the dominant shape as against bacilli and cocci. The result was dominantly catalase positive and oxidase negative. Next slide. And then higher percentage of the isolates exhibited biofilm, which shows the presence of aerobic bacteria. And then the most dominant type of hemolysis testing that was gotten was alpha hemolysis, that's the partial lysis of red blood cells, followed by gamma hemolysis, and then beta hemolysis. Next slide, please. 4 classes of antibiotics were used: the sulfonamides, the carbapenem, the penicillins, and the cephalosporins. We got the presumptive bacteria were gotten via the shape, from the shape, Gram-Stein reaction, and hemolysis testing results, and 7 presumptive bacteria were gotten. The result of antibiotic susceptibility testing was compared with the clinical and laboratory standard institute, and Corynebacterium species, Listeria monocytogenes, Lactobacillus species, Bacillus gram-positive rods had no breaking point on TLSI, and Enterococcus species was susceptible to sulfamethoxazole and doxycycline. Streptococcus species was susceptible to sulfamethoxazole, doxycycline, and imipenem, while Staphylococcus species was resistant to all antibiotics. Conclusively, this study demonstrates that external surfaces of these selected canned drinks are they have high population of bacteria, and the presumptive bacteria were all pathogenic, and more bacterial isolates were gotten from surfaces of non-disinfected canned drinks than rinsed or disinfected ones. And these pathogens are antibiotic resistant. They are resistant to antibiotics, and they cause microbial resistance in consumers of canned drinks. Next slide. How do you now take your canned drinks? Safe consumption practices should be adopted. You disinfect the surfaces of canned drinks. In absence of disinfectant, make sure you clean the surfaces of your canned drinks with tissue or clean handkerchief, or you use newly unwrapped straws to take your canned drinks. Public sensitization and awareness should be adopted through media and community outreaches, and the regulatory system should be strengthened to protect the public health. Thank you.
Thank you. Thank you. Thank you. We really appreciate our presenters for sharing their research findings with us, and we wish you all the best in your future endeavors. You can go back to your seats. We would like to call on one more presenter from SQ High. They are one of our sponsors, and I've been instructed to give them that opportunity to present with one of their findings with us. I would like to call on Ojo. Okay, you have the floor. You have 5 minutes to tell us about this beautiful work.
Thank you so much, Ma. My name is Rosalie Moronkola. I'm representing Mr. S.O. Ojo. And the title of this brief presentation is Multidisciplinary Pain Approach in Artificial Intelligence as a fundamental drive for biomedical research. The aim of the presentation is to connect pain mechanism in biology and AI using reinforcement learning and ethical modeling. I want to show you how biology has been applied in artificial intelligence, which most of the presenters has actually touched a part of it, but we may not notice, and I'm going to expose us to what it is. Pain mechanism is a feedback mechanism that prevents harmful actions, especially when we are children. As a child, when you don't need to tell a child not to touch fire. Once he or she dips his finger into it, it sends a signal to the neurological system, which tells him or her that this is an harmful action, and the child will invariably not try that action again. This ideology has been applied in artificial intelligence also, in order to make sure that our artificial intelligence agents predict accurately. Some of our presenters have presented some findings in which they built models using logistic regression, support vector machines, random forest, and one of them also used artificial neural network. Most of these are pain and error in machine learning. How does pain mechanism link with artificial intelligence? This is done using loss function, reward systems. Artificial intelligence models or machine learning models uses the idea or the strategy of loss function in order to make sure that these models predict accurately. For example, in the prediction of COVID-19 status of patients, a study was carried out in which a machine learning model was developed in order to predict the COVID-19 status of patients. This model helps to reduce laboratory testing time. Instead of the patient going directly to the laboratory in order to check their status after the result of the lab test, these AI models can easily detect or predict their diagnosis status, whether positive or negative. But how will you assure the accuracy of these models so that somebody that is COVID-19 positive will not be will not be left untreated, and someone that is positive will not be left untreated, and the person will actually die of the sickness? And to prevent wrong diagnosis in essence, that is why pain mechanism has been incorporated into artificial intelligence in the form of loss function. This loss function ensures that the model predicts accurately, and this is done by penalizing the model, in which the common penalties used are L1 penalties and L2 penalties, which are lasso penalties and ridge regression penalty. This has been applied in reinforcement learning, which is a major aspect of artificial intelligence framework. Negative rewards is being assigned to the AI model when it took the wrong action. That is when wrong prediction is taken. This error or loss value is the difference between the actual prediction and the actual value and the predicted value. That means that is the patient actually positive for this disease, and what is the prediction of the model. So the difference in this prediction must be minimal so that the life of the patient will not be put at risk. So this pain mechanism helps the machine learning model or the AI model to be able to take the right decision in order to ensure accurate prediction. Adversarial trainings are also one of the ways in which pain mechanism has been incorporated into AI. AI system also dictates pain from facial expression. We have AI model that can detect your facial expression. They can detect your facial expression to know whether you are actually feeling pain at that time, based on maybe you squeeze your face, based on how the facial features will be captured, and the model will be able to predict such. Ethics and human-centered AI. Embedding artificial pain can enhance AI safety, but raises moral questions about empathy and anthropomorphism. Future direction. Bio-inspired loss function. We are to make sure that your AI model predicts accurately. There must be custom loss function in which you design your loss formula to suit the field of study in which it is being applied to. AI can also be used for personalized pain medicine, and ethical AI pain frameworks are also areas that can be explored in the future. Conclusively, pain principles guide both biological and artificial learning. Integrating them improves adaptability, robustness, and ethical alignment in AI.
Thank you. Please, you can drop it. We appreciate our patience, and we have come to the end of the oral presentation session. Thank you very much.
Ladies and gentlemen, please a round of applause for Dr. Mrs. Ajayi as the moderator for the oral presentation. Like she said, thank you so much for your patience so far since morning. I can remember when we started, the atmosphere was clear. Now it's getting a little bit dark. And we still have a lot of people with us in the hall. Please, can we take time to celebrate ourselves? Put your hands together for yourselves. This is commendable, really. The first Next Generation African Biomedical Science and Innovation Symposium, the first of its kind, as proudly powered by Helix Biogen Institute. We are coming to the end of that for today. I assure you we've made history together here today. I have been part of this. Excellent Excel has been part of this.
Oh, yes.
And all of us here, we have been a major part of this. And I believe everything that has been discussed here, we are going to leave this place to take actions. Thank you so much to the guest speakers. Thanks to the chief host. Thanks to the guests of honors, the invitees, the attendees, all of us that are here today. Thank you so much. And of course, Helix Biogen Institute, the Director of Science, Associate Professor Kola Oladipo. Please round of applause for him. He has done really well. All right. Before we do the closing remark, just like Professor Julius, the chief host, just like he promised, I have 10 products, Trino IB products. We have 10 for the first set. We have 10 to give away. If you are sure that you can answer a question about the symposium, I need 10 people that want to come out voluntarily, then I'll give you one product. Just these are questions that about things that happened here today.
Yeah. It's nothing big at all. So if you are confident in yourself that, oh, I think I listened attentively, or if you're confident in yourself, please just raise your hand.
Just, please get me the products. Hello, please get me the product. Just get like 2 or 3. Like 2 or, no, just bring 2 or 3. 2 is okay. So this is trying to, please come forward, come out. A round of applause for them as they are coming out. A round of applause. Okay, I think we can have everything here. I think we can have it all here so that. Another one is just like the Director of Science is telling me, yes, the microphone, thank you. We have 10,000 Naira recharge card for somebody here today. If you are going to be the lucky winner, Prof, wait, sir. We have 10,000 Naira recharge card. If you are going to be the winner, I'll let you know. I'll just call out the number. Please, if you are the winner of the recharge card, please, you let us know up here. The first question I will ask, please come forward, come forward. A round of applause for them. You can join them. Oh. If you want to be part, you know you can. Please come forward, come up. Let's come up here. Okay, thank you. Why are we not having people at the side? This is free product. Oh. I personally have used Trino IB. It boosts your immune system. A lot of things that this product can do in your body system. Okay, we have 1, 2, 3, 4, 5, 6. We need 4 more. 4. àwọn obìnrin like nǹkan ọ̀fẹ́. Look at them. Look at them. Okay, ọkùnrin náà dẹ̀ wá. Okay, 1, 2. Ọkùnrin dẹ̀ wá wà ń bẹ̀ o. Kò sí ọkùnrin. 1, 2, 3, 4, 5, 6, 7, 8. 2 more. We should go with that. Okay, so I'll just ask the question. If you don't know it, that means the next person that knows it will respond. Now, what is the theme of today's symposium? Don't look back. Don't look. You can't look down. You can't look back. What is the theme of today's symposium? Please help me give them the microphone. What is the theme of today's symposium? If you know, you don't. Do you know it? Okay. What is the theme? Why are you holding this program? Are you sure this is on? Please get him this. Hello. Okay. Yes.
First,
Mm-hmm.
Nextgen,
Mm-hmm.
biomedical.
Okay. No. Let him. Don't put his head on the spike yet. Let him finish. Okay. First Nextgen. Which one are we sticking
First,
to?
Nextgen, Africa,
Yeah.
biomedical science, innovation, symposium.
And innovation symposium. Please round of applause for him. Let's. Wow. Oh, sorry. You did not get it. Oh. Thank you. You guys are wonderful. That's not the theme. In full. Okay. They said they want it in full. That's not the theme. Yes, that's not the theme. I understand what you are saying. That's not the theme. You don't know the theme. This is the title of the symposium, but this is not the theme. What you have said is not the theme. Are you ready? Okay.
Empowering the next generation through empowering the next generation through global global kingdom biomedical.
Global kingdom biomedical. What are you doing? Okay. Do you know it? Please give it to him. Okay.
Thank you. The theme for the event is empowering the next generation through global impact for young leaders.
For
young leaders.
For young leaders. People, you people, yeah. What are you doing? Give it to Anna. No. Okay, that's not okay. Please let it out.
Empowering next generation of scientists to drive global.
Are you looking at it up there?
No, in biomedical.
Biomedical
innovation.
Wait, let her take it again. I need to be sure.
Empowering.
Please face them. Okay. Yeah.
Empowering the next generation of scientists to drive global impact.
Yes.
In biomedical innovation.
Empowering the next generation to drive. Wait now, I have temper. You people want to collect it. Me, see. I want to be giving it to them. Okay. Wait. Now, for real, she has really tried. Empowering the next generation to drive global impact in biomedical innovation. Thank you. That is from Professor Julius Kolawoloke. Of course, you can go. Thank you. The next question. See, I already mentioned the next question. I was going to ask what is the name of the chief host for this event. You are still frowning your face like you are trying to remember the name of the chief host. Okay, now, but I won't do that. Helix Belgian Institute was founded in what year? No, please, audience, let's help. Mami Adechawa, what's going on? No, how many people are staff? If they are staff, you cannot respond. Are you a staff? Okay. Give her the microphone. What year was it?
2021.
2020. She's not a staff, truly. No, she's not. What year? You don't know? No, I know you. It can't be you. I know you too. Are you sure? You are a student? Okay. Let me use my microphone. What year was it founded?
2019.
2019, that's correct. Please round of applause for him too. Come and have a product. Thank you. Okay. And now, The other time, I announced it twice. The co-compare, she announced it once, too. How many abstracts were received for this symposium? How many? Look at all of them. Huh? Okay, let's give the first person.
113.
113. Are you? Okay.
118. 118.
108. You okay? Because you have the microphone, you're not. Let's give it to him. He said it. All right. The two of them. Hello, sir. He does not want it. Please, one for you and one for you too. Please show. Take picture with your IB. Please, round of applause for them. And again, what will be my next question now? Okay. So, what is the name of the keynote speaker for this symposium? Wait, be coming down. Professor, no, you have to say the full name.
I tried.
Yeah, you have tried, but you have to say the full name. That's a very simple question. Who? Do you want to try? Okay.
Professor Tomori Oluwale.
Oluwa, show me love. Oluwale Tomori. No. No, it's not Oluwale. That one, I'm not giving it out for free. Oh yeah. Okay. Are you an Igbo person?
No.
Okay, say it again. I want to hear.
Professor Tomori Oyewale.
Oyewale. Let's give it to her. Thank you. Please take picture with the product so that. And thank you so much to Professor Julius Kola-Oloke, our chief host. God bless you, sir. Okay, you want to ask them a question?
Yes, and
See, my own questions are simple.
MC the face, what do you mean? During Professor Julius's speech, he actually mentioned one thing. He said, what is that? Okay, so what is that thing that he mentioned? That he said you have to know before you can actually kickstart anything innovation, like it's foundational. I'm going to give you an hint. It says something type. Something type. That you have to know dash type. Your dash type before you actually start.
Oh, that the chief host mentioned. Give it to her. She said it. Yes, she said it first. Give the mic to her.
Enneagram.
Your
Please,
Enneagram.
can we celebrate her?
Please, round of applause for her. Please come and have one. Thank you. Take picture.
Okay.
It's mine now, Elijah.
One of our keynote addresses today, it was focused on education. So what SDG addresses education?
Someone give her the mic. Give her the microphone.
SDG 4.
Please round of applause for her. She gets it right. SDG 4. You said 14. Okay.
Okay. This other one is about SDG. What SDG addresses biomedical innovation? Biomedical innovation.
Yeah, biomedical or
what
health.
SDG? Yes.
Yeah.
Simply put, health.
No. Okay.
That's a bonus question.
That's a bonus question, really. 7. You are still guessing. Okay. Well, if now we have oral presentations here today, and we have announced before we started the presentation, how many numbers did we pick for the oral presentation? 2020.
We're not giving anybody.
Is it 20? 21 later. Now you have become. I like the way you are explaining. You are explaining with passion now. Oluwa, I go you go for me. Okay, give to her and the other person too that said 20, and of course the last lady standing. Do you have a question for her? They have a question for her. Wait, for the fact that you have come out to no. I want the 3 of you to take the picture together. Please, round of applause for them.
Let's celebrate
a round of
this
applause for them. Thank you. Now, please, if all the volunteers, we are going to wait here. After now, once we are done with the closing remark, we'll take the closing prayer. Then while people are going, volunteers, please wait. Together with some of us that paid for the cocktail, we're going to have the cocktail event. If you paid, I'm sure we have the tag, we have the card with us. So every other person can leave immediately after the closing remark. But again, before we do that, I would like to say, okay, I was saying 10,000 naira recharge card the other time. Are we ready for it now? Should I call the number now? Okay. Which one is yes? Do you want to know too now? You're already pressing your phone, Exelo.
I'm pressing
Okay.
I'm just answering on their behalf.
Okay, you're answering on their behalf. Well, no problem. I think during the cocktail event, we are going to give more products out during the cocktail
Absolutely.
event. Thank you so much.
So, ladies and gentlemen, truthfully, it's actually been a beautiful ride with us here today. I want to believe that, when I started, I said something that I hope everybody here is going to leave this place richer intellectually and also rich. I even wanted to ask one of the volunteers earlier, and richer connection-wise. I wanted to ask them that, who did you meet today? And you're going to confirm the information of that person. And meanwhile, you must not have known that person prior to now. So I really want to believe that we have connected, we have learned, and we are so ready to turn our ideas, our innovation, our discovery to actually serve humanity and to make impact globally with it. Thank you so much for coming.
Thank you. It's been an amazing evening, an amazing day with you all today. My name is Motunrayo Yode, they call me MC Face, and I've got...
I am Excel Adedokun, your co-moderator for this extraordinary symposium.
Now, for the closing remark, I would like to call on our mommy, our mother, Dr. Mrs. Oloke. Please, a round of applause, ladies and gentlemen. Please, you can do better than that. Put your hands together for mommy. Then we'll take the closing prayer by Reverend Sam Akonde.
Wow, thank you so much. We have stayed so long here, and when the moderator started, he actually started with the first thing too that I jotted down. I said we waited till now. Wow. And I said we should clap for ourselves. Please, let's do it again. Thank you very much. The abstract presenters, I really appreciate you. If you have not done anything like this before, I think you have learned from them. It's not easy standing and presenting things like that. So let's clap for them too. They really did very well. Then, I want to say that I have actually become a biotechnologist, a microbiologist. I'm a sociologist by training. But like in the Yoruba land, they say if a sponge stays long on soap, that sponge becomes soap too. So I've rubbed minds with a lot of the colors, like the host said earlier on, and it seems I'm becoming part of them. So we are really blessed, with Helix Biogen Institute, and I pray God will take you higher. There's one challenge from what we have said today. We know there are no finances, but I want to say, please develop yourself. Try to develop yourself. And then, finally, from all that we have said today, please, don't let us put them on the shelves or on our tables. Let's turn them to products. One of the presenters, at the end, she showed us something that she had made from her work. I want to challenge all of us too, as has been said, let's go out to be solution providers. Let's turn our work to products and let's make it useful to humanity. Once again, I want to say congratulations to Dr. Kola. Oh, professor on the way. I like to call him professor. Professor Kola Oladipo, Associate Prof, for this beautiful vision. And I'm praying that God will expand it in him in Jesus' name. For every participant and all attendees, some have left. I want to say congratulations to all of us. What we have learned today will, is it mutate? Or how do you say it? They will grow and break and expand and be useful to humanity. Once again, congratulations. Thank you very much. And everybody, thank you for waiting.
Please round of applause for Dr. Mrs. Oloke. Thank you, ma'am. Okay, so I promised 10,000 recharge card from Associate Professor Kolawole Oladipo. Please, just a minute. I need to be sure he wants me to call this. Okay. All right, ladies and gentlemen, in case you are going to load, if you are using MTN, Glo, or Airtel, you are going to load with star 585. Are we ready?
Regardless of the network.
Regardless of the network. Star 585 star. It's 10,000 recharge card. Please, if you are the winner, please you signify after.
So
Star 585 star. Are we ready? Okay. 91. 91. Have you pressed that? Okay, maybe I should call the second one. Let me call the second one. It's actually 10,000 naira recharge card. So let me call the second one first. Okay, the second one is 70. 70. You have pressed that. 37 79 71 15. I've called 70 37 79 71 15. Ladies and gentlemen, all you need to do is where we started the first 7, go and put 0 there. It's going to give you 070 3779 7115. That is my phone number. In case you have an event, you want me to anchor, you want me to be your host, I don't charge much. My name is Mutrayo, you're the MC of Face, and it's been a wonderful evening. Thank you. God bless. I love you all.
Thank you for sticking with us.
For the closing prayer, please, Reverend Sam Akonde, for the closing prayer, please. Okay, it's not there. Okay. Okay.
Sorry. Face. Well, look, I'm going to give you.
Oh, you were pressing.
Look. You have to watch your back when you're going on the streets. I'm serious, sir, because I want your to face is marked already.
The security personnel, are they still outside? Okay. For the closing prayer now, we can as well just take somebody. Dr. Oyeru. Please, for the closing prayer, we'll take Dr. Oyeru for the closing prayer. Please round of applause for Dr. Oyeru as he's coming out for the closing prayer. Thank you. He works together with Associate Professor Kolawole Oladipo at Adeleke University. Thank you. If you know you are not more than God, please let's rise up while we say the closing prayer together.
Yeah. Please let us rise. And I would like us to sing this song in Yoruba. Adúpẹ́, adúpẹ́ lọ́wọ́ Ọlọ́run nítorí bó ṣeun àánú rẹ̀ yóò wá títí. Adúpẹ́, adúpẹ́ lọ́wọ́ Ọlọ́run. And so, Father, this is our confession that all glory belongs to you. Thank you for the wisdom. Thank you for the grace. Thank you for the enablement. Thank you for the start of this program. Thank you for the middle. Thank you for the end. Indeed, you are the Alpha and the Omega. We return all glory to you. Receive our praise in the name of Jesus Christ. Thank you for every sponsor. Thank you for the chief host. Thank you for the co-host. Thank you for everyone in attendance. Lord, we give you all the glory. Receive our thanksgiving in the name of Jesus Christ. We thank you for journey mercy you have already granted to those that came, and everyone here that will still be on their way right now. Lord, we ask that your presence be with them in the name of Jesus Christ. Thank you, everlasting Father. And so we declare this meeting open. We declare it closed in the name of the Father, and of the Son, and of the Holy Ghost. In Jesus' precious name we have prayed. Amen.
Thank you, ladies and gentlemen. That will be all. See you at Nextgen Africa Biomedical Science and Innovation 2027. 2027. We are coming back bigger, better.
I'll be there.
I'll be there. Yes. Tell the person next to you, I'll be there.
I'll be there.
All right. We can take our leave now. Please, all volunteers. All volunteers, let's wait behind. And those people that paid for the cocktail,